Hi, welcome to Detours, Understanding Acquired Brain Injury. And this is Unit 2, Chapter 5. And this is going to be divided into two parts. So we'll be doing Chapter 5 will be Part 1, and then Chapter 6 will be Part 2. We'll be talking about meiosis, or the talk, because this is going to be about how sex cells are produced this time. Let's talk about sex cells. And this is another process related to cell division, and we call this meiosis. Some specialized cells are devoted to the production of an entire organism. And that's what's different in this case. Unlike other cells, sex cells are needed to reproduce the entire organism. And whether that is an animal, whether it is a plant, fungi, other things, even some bacteria reproduce sexually. And sex cells are commonly referred to as gametes. And these gametes are produced in specific organisms of the animal, the plant, whatever, and they're called gonads in animals. Gonads reach maturity when the organism is physically, at least, mature enough to provide care for the young. Now, obviously, some animals, some plants take longer to reach that state than others do. Now, the dividing of these gametes is called meiosis, as I said. And it evolved basically to increase the chance of survival, to combat genetic errors due to a lack of genetic variability that happens in eukaryotes over time and can lead to genetic mistakes and things like that. So that's why these cells divide in the way that they do. So the first way to fight the risk of genetic errors is a process called crossover. Crossover is important because it blends genes from maternal and the paternal line in the organism that is producing the gametes. So when we make our gametes, we undergo crossover. And so our parents each gave us different sets of chromosomes. And each pair of chromosomes will undergo at least one crossover. And so this exchange is important because it kind of shuffles some of the genes around. Now, there are each individual chromosome, when they pair up, you'll get the sequence from both, and it is this exchange that will, this crossover that will take place. And it primarily happens, and we'll get into details, in the areas where the chromosomes do the same kind of job. And so that decreases the chances of mistakes happening when it comes to crossing over. This exchange, we refer to these areas that have the same function as homologous chromosomes, or homologous regions of the chromosomes. The exchange, when it comes to these crossovers between these homologous regions, explains why a child you have may look more like your mom or your dad, or like your maternal or paternal grandparent or something along those lines, because segments of chromosomes have been exchanged in yourself or in your partner that come from their parents in turn. And so that's why. Let's get into the mechanics. Okay. Crossover is a process that's based on a repair mechanism. At least that's what evidence suggests. It's just that it became used for a different purpose when it comes to the production of gametes. In the cookbook analogy, family members might swap cookie for cookie recipes or cake for cake recipes, but they're less likely to swap a soup recipe for fruit cake, for example. And this is what I was talking about when I said homologous areas in the chromosomes are most likely to swap with each other. So let's look at the steps of how crossover happens. It happens typically at the end of prophase 1. And these basically how a cell divides, becomes a sex cell. It goes through 2 passages through the cell division. in order to reduce the number of chromosomes that are present in any sex cell, because you've got to combine information from mom and from dad together in order to produce a new individual. So you'll be hearing me use the term 1 and 2 when it comes to how cells are divided. So first, the paired chromosomes will line up to form what's called a synaptosomal complex. So, and this is inside of you or your partner or whatever, long before we're going to be putting sperm and egg together. So this is how your sperm or how your eggs are going to divide up, so we get the crossovers that we need. So first, the paired chromosomes in you or your partner will line up and to form this synaptosomal complex, which is a group of these homologous chromosomes. One from mom, one from your mom, one from your partner's mom, one from your partner's mom and dad. Okay? Here. Okay. The chromosomes that cross genetic material are not sister chromatids because they are not identical copies. Remember, we haven't made a copy. We are lining up, one's from mom, one's from dad first. Then we are going to exchange materials using the synaptosomal complex. The synaptosome will hold together the non-duplicated chromosomes. Then they will exchange segments of genes. They're basically these chromosomes will be cut, and there's a set of enzymes called recombinases who will basically pull one pair to the opposite side of these non-sister chromatids, of these your maternal paternal chromatids, over and swap them. Exchange will occur between non-conserved regions. We've talked about that before, that there are certain regions that are so important we can't risk making errors there. Things that control symmetry of the body, that make sure you develop eyes, things like that can't be exchanged. You need to make sure you have eyes, or make sure that you have a left and a right side. Do things sometimes go wrong? Yes. And most of those, when that fails, leads to a destruction of the cell. But sometimes things happen, and this is why there's a miscarriage very early on when something malfunctions here, and why there are a lot of early miscarriages, because serious malfunctions occur during, say, crossover. This exchange, where genes are in the process of swapping, it forms what's called a holiday junction. Like Doc Holliday, that kind of holiday. Holiday junction, where the chromosomes are cut and they're pulled. It looks almost like a four-way intersection as chromosomes are pulled across. Obviously, this happens because this is with the sex cells. This happens with all your different chromosomes, which have to be swapped as you're preparing to create sperm or eggs. So all your different pairs, all 23 pairs, are going through this process simultaneously, because you're going to be creating eggs and sperm, and you need to randomize as much as you safely can and swap around. So cuts made by the enzymes in individual segments are interwoven with each other into these holiday junctions that are formed throughout these open segments of the chromosomes. After breaking the DNA segments and the help of the RNA that exchanges the material, you have new chromosomes with new DNA sequences that will differ from you or your partner. So you've got new genes, new chromosome segments, and they're in the homologous region. So these are areas that do the same thing. Again, remember me referring to cookie for cookie recipes and soup for soup. So these areas still do the same kind of job. Skin, color, texture, or hair, or whatever. it still will exchange for that. It just will maybe be from one person's side, your mom's side, your dad's side, and swap for the new individual. Finally, recombination. The visible microscopic evidence of places where recombination took place. And this is called a chiasma. And here's a picture of one. You could see where the swap happened. And after these repairs are made to where the crossover took place, you get separation, the recombination is done, the synaptosomal complex gets broken down, and the sex cell then can complete focusing on division. Obviously, this is a very simplified way of describing this process and the steps, and there are steps inside of steps inside of steps. But we wanted to stress how important crossover is in order to provide genetic diversity for the process of cell division in meiosis and getting ready to make sure that there's a lot of variability in the genes when you pass them on to the next generation. Now, when we get into the second video, talk about meiosis as far as production of sperm and eggs. Now we know that we've got more kind of randomizing. Now we can make the sperm and the eggs knowing that they'll have different genes in them. All right. So I know this can be a bit confusing, but crossover is important to prevent the problems that you get if you just used mitosis to produce the next generation. If you just were a clone, if your son or daughter were just simply a clone. And this is why they're not, because of crossover. Okay? So if you have any questions, comments, anything like that, please put them below. Share anything you want to add. Please share this with anyone who may benefit. Please click like. Don't forget that bell icon. We'll see you next time. And please hope you found this helpful. Check out our Facebook group, Friends with Brain Injury. And see you next time. Bye.